This is a production of Cornell University.
Thanks, Bill. That's very nice. Appreciate it. Nice to see everybody here. So we may be in the post-genomic era, but apparently we're not in the post-video technology era. So here we are. All right, so I'm just going to jump right in and tell you that a number of questions I want to address after 50 years or so of working on polyploidy. So what does it mean to be polyploid? That's one thing. What does it mean to be diploid? That's another question related to that. Are there biologically meaningful classes of polyploids? And finally, if so, how should they be defined? Those are the things that continue to interest me. They've interested me for many years, and I'm still interested in them. So this story, it's a story today, not so much a seminar, but a story, starts with a request from the American Journal of Botany to review a paper. And we all do that from time to time, and we know that that can be an interesting experience. This was more interesting than most, and that's the theme of this whole talk. The paper was Genome Diversity and Dynamism Across the Polyploid Continuum by a really stellar group of authors. So the authors were, and you see from the left here, right here, Twyford, who is a kind of next generation behind me of people doing really interesting systematic work and synthetic work on speciation and things like that. Doug and Pam Soltis, who are probably the most prominent people in the area of polyploid evolution, along with Jonathan Wendell in the center there, holding some wine, which is one of his favorite things. And then up in the top corner is Justin Conover. And Justin was a Wendell PhD student who then went to work with Mike Barker at the University of Arizona. Mike is a really prominent person, and you'll hear all these names a lot in the papers that I talk about throughout this talk. So it's a really stellar cast of people. Now, they also happen to be good friends of mine. So Jonathan has been a friend since we were graduate students, and Doug and I go back all the way to undergraduate and to both at Indiana University. Doug was best man at our wedding. That is not his wife there, that's Jane's sister, is the maid of honor. So just so you know, that's not Pam. She hasn't changed that much in those years. Anyway, so these are really good friends of mine. And when you're reviewing a paper by friends, you can get away with stuff that you probably wouldn't want to get away with otherwise, like signing your name to the review, for example, right? So that's what I did. And this was my review, almost the beginning of the review. This is a valuable contribution by a stellar group of authors. I could stop here and simply recommend publication, but what fun would that be? So I'll provide a few reactions, some of them probably curmudgeonly or provocative or both, and thoughts that might be of interest to the authors. Now, not everybody here probably knows what a curmudgeon is, right? But a curmudgeon, here's the definition. It is a bad-tempered person, especially an old one. Now, I fit at least one of those definitions, and I'll leave it to you to decide which one you want to go with. So curmudgeon, that's what this review was, sort of curmudgeonly. And I signed it, okay, so that's the other point. So I had the guts to do that at least. So other part of the review, my review of this, the authors point out many of the problems with naming parts of what they call a continuum. But if it really is a continuum, what's the evidence that there are discontinuities that make this a useful exercise? That's never stated, and the questions at the end of the paper assume the existence of the same historical categories whose existence the authors have spent many pages and even more references debunking. So should this review not instead make the case that it's time to abandon these arbitrary categories entirely? The main point of the paper is how complex the situation is, how poorly suited to a simple one-size-fits-all taxonomy. How much more data do we need before someone decides to stop using them? And if not these authors, who in the polyploidy community has the knowledge and prestige to do so? So that was my review, and I signed it, as I said. All right, so what that led to was a response, comments from the authors, a revision by the authors that also wasn't quite what I thought it should be. And I was in touch with the editors, and they invited me to write a rebuttal to this. Right, so the plot thickens here. And I recruited Annalise Mason, who's really one of the premier people in polyploid evolution, works on brassica, among other things. And so we were all poised to write a rebuttal to this, and the authors invited us to join them and write a consensus paper. So now there's a single paper at the end of all this that's a kind of a compromise among various different points of view. So that's what I'm going to talk about today, and where we sort of end up. Annalise and I, well, we end up stuck with the term polyploid continuum, which neither of us liked that much, right? So we're now writing another paper about sort of following up on this one. So that's what's happening. So, and one of our working titles is Polyploidy in the Post-Genomic Era. And for those of you who don't know, I had to look it up on Wikipedia. I thought it was a neat sounding term, post-genomic. And there is actually something called that. It's the period after the completion of the Human Genome Project to the present day. And there's stuff about epistemology here, which I won't get into. But basically, it refers to a period where we have complete reference genomes of many reference organisms. And so as an example of what I'm thinking about when I want to know what we can do, what we'll be able to do to address these kinds of questions in the future, this paper from Faye's group and featuring Hong-Hye Sun, who is one of our own students here who just finished recently. This wonderful paper, I think it's really a beautiful piece of work, that's working on a very complex hexaploid, so 6 genomes, 90 chromosomes, phased all the way to haplotype completion. So this is what we're going to have to work with in the future as we think about how to address the kinds of questions that we've been looking at for many years. Okay, so what is polyploidy? Well, this goes back to a long way. I'm going to be referring a lot to Ledyard Stebbins' work. Stebbins was a curmudgeon, certainly. And Stebbins was one of the premier synthesizers, but one of the plant person who was involved in the neo-Darwinian synthesis, along with Ernst Mayr and Dobzhansky. So what he defines is says the most widespread and distinctive cytogenetic process that's affected the evolution of plants. It's been polyploidy, the multiplication of entire genomes. So that's the definition. And pay attention to the fact that this is a chromosomal definition. So those genomes that we're talking about are chromosome sets, and that's where we start with all this. In fact, we wouldn't have had polyploidy until people started identifying chromosomes in the late 1800s. And in 1910 or so, polyploidy was first defined. All right, so what is a polyploid is a somewhat different question than what is polyploidy. Multiplication of genomes is one thing, but who has those genomes that get multiplied, and how do you deal with that? So whole genome duplication or multiplication, we have the multiplication of entire chromosome sets as Stebbins defined it, and we have different numbers of chromosomes that you can observe. That observation leads to the conclusion that you have diploids, and then if you observe something with more chromosomes, it's likely to be a tetraploid and so on, octoploid, and up from there. By looking at lots of different plant species, Stebbins and others, synthesizing all that data, decided that between 30 and 35% of species of flowering plants and a considerably higher percentage of ferns, which I'm not going to talk about today, possess gametic chromosome numbers, which are multiples of a basic diploid number found in their genus. So those are polyploids, things that have a multiplication of chromosome sets. And more recently, people have taken even more data as more have accumulated. In 2009, Mike Barker is on this paper when he was still a postdoc with Lauren Riesberg, as I recall. But anyway, combining information, we see basically about 35% of plants that are standing, apparently polyploid species. And this is based on essentially calculated from that a 15% rate of speciation events that involve multiplication of the genome in flowering plants. Okay, so it gets a little more complicated when you have high numbers but no low numbers, right? And so what Stebbins basically says, if you look and you don't know whether these things are tetraploids, diploids, octoploids, what are they? Stebbins says basic numbers much higher than a mode of 12, 13, or 14 are almost certainly of polyploid origin. So that kind of numerology suggests there's more polyploidy out there than just what you get if you just do the numbers of extant species in populations. So take us to the genomic era now, as genomes start to appear. And this is a seminal paper looking at gene duplications. Well, if you don't duplicate sets of chromosomes, obviously the genes on those chromosomes are duplicated. And if you look at the divergence between those duplicated genes and estimate how old the duplications occur, that's what this slide is about, work from Lynch and Connery. And so what you see if you look across species, humans included, what you see are cases where what's happening here. Aha. Wow. My slide didn't do what I thought it was going to do. Okay, wait a second, sorry. All right, let me try this again. Okay, there are small scale duplications of genes. You see a lot of genes that are almost identical to one another in pairs, right? And those genes apparently duplicated very recently. And so what this shows is that there are these small scale or single gene duplications and occurring very frequently in all organisms, but mostly this leads to loss because you don't retain all those genes in the genomes. You have the drop off right away. But in the background, in some species, like Arabidopsis, is a secondary peak. And this peak is a peak that occurs because genes duplicated a longer time ago, and then many were lost, but you still retain evidence of that whole genome duplication. That's the interpretation of that secondary peak. So what people then saw, and this is work from the Tanksley lab some years back now, origins of genomic duplications in Arabidopsis, this KS peak or divergence peak that you see on the right, but also then looking at synteny of these genes, they occur in blocks. And so blocks of genes have been duplicated. Originally whole chromosomes is the interpretation. So there are whole genome duplications. This is a seminal paper from Blanc and Wolf from 2004 explaining how this works. You have this initial peak of duplication, exponential decrease, but then you see these secondary peaks based on whole genome duplications that decay over time as well, but with a different rate than these initial duplications of individual genes. So Blanc and Wolf and others, we were involved in some of this work, looked at the distribution of these KS peaks, these duplication peaks across many different species and found them all over the place. So there's a lot of duplication going on. Now, to confirm that duplications are shared, you've got to do phylogeny, to look at gene relationships and see the different patterns that you expect to get if a duplication is shared among different species. So that's something else that had to happen for this all to become interpretable. But basically by 2011, it was discovered that all flowering plants have experienced a duplication. In fact, all seed plants have, and then flowering plants have experienced a second duplication as well. So there's a polyploid ancestor already, and the angiosperm ancestor experienced a second whole genome duplication. Jonathan Wendell wrote a very nice little review in 2015 showing at that point from the genomes that had been sequenced how many duplications, whole genome duplications, had been inferred from those genomes. So there's a lot of stuff going on here. All those little squares are duplications or even triplications. And so if you start with an ancestor that was a diploid back then, all the way at the dawn of the seed plants, and you have a duplication in seed plants, take it to a tetraploid, an octoploid, a triplication here in the eudicots leading to Arabidopsis and soybean, the arrows pointing to soybean, which I've worked on. So 24x by there, 48x at the beginning of the legumes, and then 96x in soybean. So not a diploid, but a 96-ploid plant. Arabidopsis, what about that? Well, notice the two duplications at the beginning of the brassicales there. So Arabidopsis, although it only has 10 chromosomes and of course a small genome, is actually 2n equals 96x equals 10. So there's a lot of polyploidy going on here. All right, and this is one reason why so many plant gene families are very complex with lots of members. So if you think about what's happening here, if you started out with one copy and had a duplication and a second duplication and a triplication and then speciation and more duplications, this is what you get. That's what a gene family would look like just based on polyploidy in soybean and not even soybean, there's one more duplication in soybean. So this leads a lot of people to be pretty, well, not happy about polyploidy. So for example, we have, so this is, I got her permission to say this to not the cartoon, but just the AI helping with the cartoon, but it's not a very flattering picture. Anyway, but it does complicate things quite a bit to have polyploidy. And that's a direct quote, by the way. I just want to say. All right, so this is a very nice paper on gene families. And considering that at least 5 rounds of whole genome duplication took place, and that's on one estimate, if you had 10,000 genes in Arabidopsis, the number of genes should now be about 320,000. And that's even without additional small duplications that occur. And obviously that's not the case. We don't have that. So what happens? It's about 10 times higher than we actually see. And although some duplicates have survived over millions, or hundreds of millions of years, the predominant fate of most duplicates is loss. And it's mostly loss we think of as pseudogenization. So pseudogenes are formed, followed by loss of these non-functional genes. So it's pretty typical. And we'll see this, I'll call this fractionation. This is what it's called primarily in the polyploidy literature. Okay, so here's the rest of Jonathan's really nice picture and explaining what he means by wondrous cycles of polyploidy. So you've got all this polyploidy going on, but what happens, and he works on cotton, so that's what you see here on the right-hand side. You have these genes from two different genomes that are combined in cotton, and they experience different fates once they're combined in the same nucleus. And so we see changes in rate and recombination rates. Transfer among the genomes can happen, subfunctionalization and neofunctionalization, new functions for these genes, all kinds of things. And down at the bottom, this cycle of where you have one genome and then two genomes together through whole genome duplication. There's gene loss and then biased fractionation, a phenomenon where one genome loses more genes than the other does. And then downsizing of the genome that can happen, reducing numbers of repeats, transposable elements, things like that. And then rearrangements, loss of centromeres, and then you end up with something that looks, for all intents and purposes, like a diploid, and then the process starts over again with the next round of polyploidy. So that's the cycles we're talking about. And the process is called diploidization. So you start with a polyploid, and it becomes more and more like a diploid over time. That's diploidization. So here's a nice paper from the Barker lab, nice review from a couple of years ago now. Patterns and processes of this diploidization process. And so in the most basic sense, what it is a return of a polyploid genome to a diploid state. So diploidization adds a temporal component to this polyploid continuum that we're talking about in this review paper. So basically, it's the diploid polyploid continuum is one way to think about it, okay? And then we have this landscape of genomic variation. So diploidization, return to from polyploidy to diploid state. There are many mechanisms that contribute to this, and they can broadly be described as being independent from one another as cytological diploidization or genic diploidization or fractionation. So fractionation is what we're talking about here for starters. So are diploidization mechanisms specific to polyploids? Is there anything special about what happens to a polyploid versus to a diploid? That's the one question. Does diploidization have rules? Do all polyploids follow the same kind of processes? So here's fractionation. We'll start by talking a little about that. So Yves van der Peer is another person who's really made major contributions to the polyploidy literature and understanding. And the paper just came out in the end of the summer, I guess, subordinate role of pseudogenization to recombinative deletion following polyploidization in angiosperm. So that same diagram I showed you before of pseudogenes leading to gene loss, well, it turns out what they're showing in this paper, is that in that process is true in diploids primarily. So you have pseudogenes accumulating random mutations and becoming inactivated when a gene is redundant, right? So it can accumulate those mutations, and then eventually it's lost from the genome. Whereas what they're seeing is that in polyploids, it skips that pseudogenization process, but goes right through recombination to deleting whole genes. And they see much more of that in many, many genomes that they've looked at in the process of writing this paper. So that would be one way in which perhaps there's something special about polyploidy in these mechanisms that are leading to diploidization. So another question, of course, is how does a duplicated gene escape the fate of being lost or even inactivated? And so there are many mechanisms where this can happen. Some you're probably familiar with. If a gene accumulates a new function, gets a new function, or changes its function somewhat by neofunctionalization, subfunctionalization, you've probably heard those terms, a gene can become indispensable so that both copies are retained, and that's a pretty common mechanism. There's also, though, dosage balance that restrains genes from being lost. It keeps them preserved. This is something that a number of people have talked about, mostly Jim Birchler is one of the main people to do it. Also, Mike Freeling has been involved in this. But basically, the idea is that dosage-sensitive genes are genes that have to maintain their stoichiometry relative to the things with which they interact. So we've got genes that are involved in various different networks, for example, where the stoichiometry may be important, but also multi-protein complexes that are working together so that you need to retain stoichiometry. And here we see a difference, people have seen a difference between whole genome duplication where pressure is to retain both copies, because if you lose only one copy, you may change the balance of the stoichiometry across the entire interacting network. Everything else is retained at a level, but you've lost now one of the members that's contributing to that. And that's not good. So these are deletion resistant genes. Whereas for single gene duplications, there's pressure to lose a gene that is duplicated because, again, you'll change the stoichiometry. So we can see this, and we've done some studies in the soybean polyploids, recent polyploids, looked at those genome-wide over many different genomes, see the pattern of retention of particular classes of genes, Yves van der Peer, again, some of his work. And also we can see that this happens very rapidly with transcriptomes showing the effect of dosage even right after a synthetic polyploid is actually made. So there's, again, a difference between diploids and diploidizing polyploids in this area. So are there different kinds of polyploids with different rules? This is the other topic that I want to talk about. It was a major topic in that review paper. So the processes of diploidization involve many mechanisms and forces. It's not yet clear how they operate in different lineages. Every polyploid is unique. So that's something that I think people don't think about. We talk about a class of things called polyploids, but in fact, a polyploid is generated all the time individually with its own set of circumstances. And as they point out here, post whole genome duplication lineage experiences different demography, selection pressures, other population genetic differences that could drive unique rates of gene loss and other phenomena involved in diploidization. So if we think about diploidization as bringing us back towards the diploid level, we look at this over time. I just drew these little diagrams. Diploidization process may be lineage specific. So if you think about the same process, different species may do it differently. Some with, on the left-hand side, a big drop off right away down back towards the diploid condition, and then a decline after that. Some that don't drop as much, and others that are much more gradual. And this can be, different species can be at different stages of the same process. So even if the kind of response curve is the same between two species, because polyploidy is happening all the time, different polyploids are of different ages and maybe at different stages. And also, different processes have different responses, even within the same species. So cytological diploidization may not be occurring at the same rate as fractionation of genes, for example, right? And so we see this. This is work of a colleague of mine in China, in Beijing, Shenwei Zhu. And she and her group have shown that, as this paper says, diploidization in a wild rice allopolyploid is both episodic and gradual. The polyploid is down in the middle, down there in the bottom left. And this is a species that's about 700,000 years old, estimated from the molecular rates of gene evolution. So what do we mean? Well, gene loss by fractionation has occurred in an early burst, it looks like, in this, by looking at multiple individuals of genomes of this particular group. But in contrast, the accumulation of transposable elements and also homologous exchanges by recombination seem to be much more gradual and based on the demographics of what they can look at there. So if we now think about this, the diploid-polyploid continuum is a multidimensional landscape of polyploid genome variation. And so we ask, are there different kinds of polyploids with different rules? If there are, the question is really, is the diploid-polyploid continuum, whose endpoints have been called autopolyploid and allopolyploid, a multi-dimensional landscape of genomic variation? So that's quite a mouthful, and you can see why that was not the title of the paper originally, although it maybe should have been. So let's go back again in history, and now we look at when polyploidy was first discovered. Within a dozen or so years of that, people were finding that there were two classes, or named two classes of polyploids, Kihara and Ono, in the 1920s. And here, for those who don't read German, basically autopolyploidy is doubling of the same chromosome set, whereas allopolyploidy is the coming together of different sets of chromosomes through hybridization resulting in chromosome duplication. So allopolyploidy is hybrid polyploidy, and autopolyploidy is non-hybrid polyploidy by these definitions. And to take it a step further, that hybrid polyploidy for most systematists, most of the people looking and asking about whether or not the plants that they're working on are auto or allopolyploid, it involves two species if you're an allopolyploid and only one species if you're talking about autopolyploidy. So both of these involve genome duplication. So they have two sets of chromosomes, right? An additional set of chromosomes. It's a tetraploid. But hybrid polyploidy, allopolyploidy, involves the merger of two genomes. All right, so what do we think about in terms of these processes? Well, okay, so we have this response curve I show on the left-hand side. Very interested what happens right after polyploids formed. And that's this stage here, the very earliest stages of polyploidy. What's the response look like? And again, is there a difference in that response? So again, I mentioned that gene balance predicts transcriptional responses that are consistent with dosage balance early in the evolution of a synthetic polyploid. Here's another synthetic polyploid. Whoop. Another synthetic polyploid. This is a colleague in Nanjing has produced these beautiful cucumber polyploids by synthetic hybridization and doubling of the chromosome sets. So what they're seeing here is that there are changes that happen in that synthetic polyploid right away. And for example, there's a shift from unbiased to biased expression of genes in the polyploid immediately in this synthetic polyploid. So just caused by the merger and doubling of the genomes. Why does that happen? Well, again, Jonathan has been very involved in thinking about this, modeling the actual transcriptional changes, the regulatory changes that lead to transcriptional differences in polyploids. It's quite complicated because not only do you have allele-specific expression as you do in diploids, but also these are coming from two different species. And so there's transregulation and cisregulation to be considered there. Genomic shock is something that's considered to happen in the earliest stages of polyploid formation as a response to polyploidy, whichever it is. And Barbara McClintock, that was her term for shock of the genome. This is a sort of review of that in the later part of her career. But it appears to be much more due to genome merger, so the hybridization component rather than the doubling. And if you read this paper by McClintock, there's no mention of polyploidy in it other than the fact that there's hybridization that's important. That's the thing that she really talks about with release of transposable elements from their sort of constraints in the normal genome. Okay, so classes of whole genome polyploidy, I've talked about this already. If you want to understand what genome doubling does, you don't want to look at a hybrid polyploid where two genomes are being combined. Instead, you want to look at what the Soltises have called pure polyploidy, and that's autopolyploidy, just doubling the same genome more or less. And so if you think about this, autopolyploidy is an epigenetic macromutation. A macromutation, polyploidy has been called that by many other people over many years, but a macromutation is a single event, polyploidy in this case, with many phenotypic consequences. Epigenetics has been defined variously, but here's one definition: molecules and mechanisms that can perpetuate alternative gene activity states in the context of the same DNA sequence. So a synthetic autopolyploid, which is isogenic with the diploid from which you created it, has many phenotypic consequences, so it's a macromutation. And it's also the same DNA sequence, so it is epigenetic, so it qualifies in both of those counts. Been interested in looking at why polyploids or how they may change through just doubling. And so this is some review that Jeremy Coate and I wrote a couple of years ago now. But if you think about what's happening in polyploidy, two things really. There's increased dosage of everything, but there's also increased bulk DNA amount. And somehow through all of that, we end up with different morphologies, physiology, and even ecology for polyploids. It's been well known for many years. So the nucleotype has been defined as the non-genotypic effects of DNA amount on phenotype. And there are all kinds of things that follow from that operate in a complex way, interact in a complex way with the increased dosage. But right in the middle of it all is a larger cell. So that's one thing we know is a rule, essentially, of polyploidy, that at least some cell types will increase in size when you double the genome content. So talking about autopolyploidy, it's a recurring mutation within species populations as a consequence of unreduced gamete formation. So unreduced gametes, sperm and egg, are being produced at some frequency in all populations as a mutation. These can unite to form either triploids or ultimately tetraploids and lead to plants in populations that are double the chromosome number, or in some cases triploid relative to that. Walter Lewis was my other mentor for my postdoc, really seminal figure in thinking about these issues of species population dynamism. The solstices again here, we're saying, if autopolyploidy is that common, have we grossly underestimated the number of autopolyploid species? And why would that be? Well, because when you make these synthetic polyploids, and presumably the same thing is happening in nature, the differences are subtle between the different ploidy levels. And so if you look at these Arabidopsis flowers, work from Adrian's graduate student, Dana Robinson, from a few years ago, you see that they look like Arabidopsis flowers, and the plants look like Arabidopsis plants. They don't look very different. So it's rather interesting to think about this. Most of the discussion of polyploidy emphasizes its ability to generate novelty, and rightly so, because there's a lot of polyploid speciation going on. But if you think about how subtle some of this is, it may be that really autopolyploidy is better thought of as an evidence of robustness to change, because this epigenetic macromutation changes everything, and yet what comes out of this is fairly subtle in the end. All right, so polyploids, do hybrids follow rules? There's a question, because I ask about rules. Is there something you can say that all hybrids do X? And I would say no, probably, because what happens with hybrids is genotype specific. So if you make a hybrid between two rice species, you don't get the same thing you get when you make a hybrid between two aster species, for example. All right, Stebbins talked about this. Hybrid allopolyploids versus non-hybrid, is that a good definition? This implies a much too narrow concept of hybridization. By focusing attention on the effects of crossing between widely different species, it neglects the even more important and far more common effects on polyploidy of hybridization between closely related species, or even between ecotypes of the same species, or I would say even of individuals within the same population, where you can have changes as well. And Stebbins said back in 91, summarizing many years of work by lots of people, any attempt to maintain a division of natural polyploids into two discrete categories, auto and allo, is more likely to confuse than to clarify a very complex situation of interrelationships. All right, so now you see why people start talking about maybe a continuum. But in the present book, therefore, these two terms will be used only as ways of helping the reader to relate the present discussion of polyploids with those in other books. So, and yet then he goes on and spends all kinds of time talking about the criteria by which he is now wanting to call things auto or allo. So he doesn't really stick to his guns on this very well. So external morphology and taxonomic key characters, he concludes the morphological criteria of taxonomists, though they must serve as principal basis for distinguishing species, are obviously inadequate guides to the evolutionary origin of many plant populations. Well, why is this important? Well, it's because the people who call things auto or allo primarily are taxonomists. And taxonomists don't make crosses all the time, don't do all those things. They just describe things on the basis of morphology. So most of the terminology, most of the decisions about whether something is auto or allo is whether taxonomists have said it involves two species making it or one species making it, whether it's a hybrid or not, a non-hybrid. So this is why I said the authors point out many of the problems of naming parts of what they call a continuum, but if it really is a continuum, what's the evidence there are discontinuities? And should this review not make the case, again, that these are arbitrary categories entirely? All right, so is it still a problem? Well, a number of years ago now, Mike Barker, in this case, and Don Levin, another major figure in plant evolution, did a meta-analysis on the relative abundance of autopolyploids and allopolyploids. So even though they said that there's a range of different things that you could call, how you could call different species, polyploid species, they were going to stick with just two categories and quantify them. that wouldn't have been a problem. It'd be kind of a useless exercise, but they chose as one of their 47 genera to look at something that I work on. So that was a problem for me because they completely got it wrong by going to the literature, my literature, yes, by God, and saying that they could tabulate what the situation was with auto or allo. So this is the actual situation. The colored things are diploids, actual diploid species, and the things in the middle, the squares, are all different allopolyploids formed by hybridization between those very good species. The problem is that all this variation is so subtle because there's this network of interrelationships that combines different morphologies and all sorts of other things. The problem that when you do that, it's very hard to give names to these things and actually come up with good key characters that taxonomists like for classification. And therefore, we haven't done that. And therefore, these all have the same name. And therefore, someone like Mike Barker and company reading the literature say, oh, it's all the same species. It must be an autopolyploid. But it's not. There are allopolyploids. So here's what we said. We wrote a response to this. And in it, we said, well, okay, so here's auto and allopolyploidy at the top, one progenitor versus two progenitors. If we look at the actual situation down at the very bottom, there are no autopolyploids. If you look back before we started working on these things and using molecular characters to figure out the relationships, there were no allopolyploids. It was all just called Glycine tomentella. In between where we are now, we've made some progress in the taxonomy, but we've really worked out a lot of the genetics. And we know that there are allopolyploids, but if you just go taxonomically, you end up with where Barker did and say that there are these autopolyploids which don't exist. So glycine has evolved in the diploidization process, in some sense, in a period of 40 years, has made tremendous strides towards being an allopolyploid, if you believe in that, but I don't. So anyway, so there's no continuum, and therefore it can't be one if it's a binary one species versus two definition being used. All right, so what's a better character? Maybe polysomic versus disomic inheritance. And what that is whether or not there is preferential pairing, as there is in diploids normally, and you will get pairs of chromosomes and nice maintenance of disomic inheritance, or if there's random pairing among four homologous chromosomes in a strict autopolyploid, you end up with random pairing and segregation ratios that don't look Mendelian, your typical diploid Mendelian ratios at all. So this is the product of polysomic, or in this case, tetrasomic inheritance. So if you could look at that, you could therefore categorize polyploids by what they're doing now and figure out whether or not they are actually auto in that sense versus allo in a cytological sense. All right, so now we talk about this. A allopolyploid has two pairs of homologous chromosomes that preferentially associate with one another and leads to disomic segregation. And an autopolyploid has four homologous chromosomes that associate randomly and produce these tetrasomic segregation patterns. So Sue Sherman-Broyles and I looked at this. We said, okay, there's a genetic definition and a taxonomic definition. So you see here, what's the situation if you put all that together? You ask, well, okay, there's classic auto and allo polyploids that agree on both counts in the corners, the top left and top right, but there are these other more gray zone sorts of things there. How well populated are those? We didn't really know at the time, and we still don't know a huge amount, but Annalise Mason, whose name you've heard already, has done an analysis of what's out there. It says our results suggest that observations of polysomic inheritance can lead to relabeling of taxonomically allopolyploid species as autopolyploid. So it looks like there isn't necessarily a very good agreement between those two schemes, which points out why there are problems with using that taxonomic classification. Here back to the more recent Barker work, the Lee et al. Paper. And so here the idea is that cytological diploidization isn't needed for an allopolyploid over there on the far left because there are already disomic inheritance and preferential pairing of homologous chromosomes. But that for a autopolyploid will go from multivalent pairing where all the chromosomes can associate together and which can lead to meiotic aberrations and abnormalities and reduce the fertility. That will lead eventually to bivalent pairing as part of this diploidization process. So for that, we maybe have random pairing going to preferential pairing over time. Now, Annalise looked at that as well. It's not clear from their results whether autopolyploids with polysomic inheritance can or do, in fact, establish disomic inheritance, and we're missing a good deal of data. We need more information on that. It's a more complicated situation because, in fact, established autopolyploids may already have preferentially done something or been the genotypes that have actually come together to produce successful autopolyploids may already have compromised genes for recombination that prevent some of the pairing from occurring among different homologous chromosomes. So there may be some preferential pairing already. So this is a very nice paper by Kirsten Bomblies talking about how that might happen. And there's more evidence of that increasing, and how this is actually happening involves introgression and the ability of polyploids to breed together and to actually cross with their diploid progenitors to continue having gene flow across polyploid levels or ploidy levels, something that people also didn't think happened much. More complication is that there's also homologous exchange, so recombinational exchange between the preferentially pairing sets of chromosomes is an allopolyploid. And this is another nice paper by Annalise and Jonathan. And here you see polyploid on the side, an allopolyploid without homologous exchanges. It stays the same, two sets of homologous chromosomes. But if there's enough homologous exchange, we can actually go to something that looks like an autopolyploid in terms of its genome structure. In between are these things that Stebbins called segmental allopolyploids. They're kind of unicorns. People haven't been able to find very much evidence of those, though there is some. In peanut, there's some evidence that maybe this kind of homologous exchange is leading to the production of things that are sort of in between an auto and an allopolyploid. A recent paper from Bao Liu, colleague of Jonathan Wendell, has looked at the rapid formation of these stable autotetraploid rice from hybrids between indica and japonica rice by promoting the number of homologous exchanges and actually converting what is in some senses an allopolyploid into an autopolyploid. All right, so I'm finishing up here. Given the present accessibility of long-range sequencing, Annalise is saying that we should be able to figure out more about how to define auto and allo and look at inheritance in different species. That's always going to be a difficult issue to do. You've got to look at segregating progeny at the whole genome scale to really figure this out. But it's technically feasible now. It will give you a snapshot of what polyploids are doing cytologically now. It may or may not help you understand whether they started that way. That's another issue for that. So here for the polyploid continuum paper, conclusions. The terms autopolyploidy and allopolyploidy are both useful and deeply embedded in the literature of polyploid origins and evolution, but classical conceptions of these terms may constrain a fuller appreciation of biological possibilities and dynamism because of perceived notions or assumptions. They retain utility as descriptions of polyploids at any one snapshot of time, but it's important to recognize, as others have previously, that these two terms represent hard to define points in a continuum or multidimensional landscape on the right there that has multiple underlying biological underpinnings and definitions, is temporally dynamic, and is taxonomically as well as genetically highly variable. So this is our conclusion for this consensus paper that we wrote. All right, so the editors also asked us to have a section on author viewpoints at the end. Okay, so each of us was given an opportunity to say what we really thought about the situation. And so here's what I said. In my experience, taxonomy does not align well with biological reality. That, together with having participated in the interminable debate on species, explains my skepticism about the usefulness of traditional binary definition of polyploids. Genomic data are confirming how the massively homoplagious or parallel independent evolution phenomenon of polyploidy can produce taxa that deal with the challenges of a double genome in many different ways and at different tempos. I was invited to join this project after reviewing the earliest version of the manuscript, which I found unsatisfyingly neutral about the shortcomings of the binary categories. Working with the other authors to achieve a consensus for this paper, I've broadened my thinking and retained my doubts. So there we are for that. So what is the hope? We've talked a bit about heterozygosity, and I want to end on this note. The distribution of heterozygosity will be different depending on whether or not you have tetrasomic inheritance or disomic inheritance. The simple reason that with tetrasomic inheritance and random assortment of chromosomes, recombination can occur across all 4 of the chromosomes in a tetraploid. And that will have an impact on the distance between the different alleles and the retention or loss of different alleles too. So looking at those patterns, I think one could, with the kind of technology that we now have and are developing now with haplotype phased genomes, actually look very carefully at what that distribution is and maybe predict where it came from, what the starting points were. That's an evolutionary question that many of us are interested in. It's not necessarily of that great importance for people who are just using the plants, growing them and doing things, for agriculture, for example. But it is something very interesting to those interested in evolution, and that's what I'd really like to be able to do. So can genome-wide patterns of heterozygosity reveal the genetic system under which a polyploid originated? That I don't know. But that's what Annalise and I are working on in our paper that we're writing right now. It is due next week. That ain't going to happen. But anyway, we're still trying to sort the whole thing out. And so those are the questions that still remain after all that has been said and done. And I want to just end by acknowledging and thanking the patience of my co-authors having to deal with me. That's a problem. And Annalise especially, who's been very good, wonderful to work with. She's very creative. And then to curmudgeons everywhere, including Ledyard Stebbins. So thank you very much. I know this was about hoping the outcome of this. Wojtek.
So, Jeff, I'm not sure if you'd agree with me, but I would argue that that using chromosome pairing as a measurement of outer polyploid or disomic inheritance, it actually may argue for more continuity than a binary division. So, for example, in tetraploid maize, which is by a taxonomic definition an outer polyploid, we see different degrees of disomic inheritance depending on the genetic background. And so if you have a polyploid inbred, it's more shows more disomic, less disomic inheritance, where you have a polyploid hybrid, the same species, would show nearly disomic inheritance. And these experiments were never done on a large number of combinations, but there's a good possibility that depending on genetic relationship of those two lines that you cross in a hybrid, you would actually see a continuum,
right,
from disomic to polysomic.
Yep. And that then you're agreeing with what I would like to see done, right? Instead of starting with something that can never be a continuum, which is to say, was one species involved or 2 species involved? That's going to be a debatable point and never going to be conducive to a really scientific approach to this. But something like pairing and the results of pairing, heterozygosity, right? That kind of thing does give us the possibility for a continuum. And there's a temporal dimension too. How long ago this either happened or ceased happening. All of those things are possibilities. And what I'm envisioning, and from the beginning, this whole idea of the landscape here, right, was that could we come up with a multi-dimensional landscape of all the things that go into diploidization and also look like they're the outcomes of how diverged the genomes are that are involved, how they're behaving cytologically, all of that, and come up with something that produced a distribution in some multi-dimensional space of these different genomes, and then ask whether or not there are concentrations of those genomes in that space that would correspond potentially to older definitions of auto versus allo. That was my idea in the first review of the paper, and it led to the landscape idea being incorporated. But that's what I was thinking specifically about whether or not you could use the same kind of approach that people use for single cell transcriptomics, basically this, and even use things like, the kind of RNA velocity kind of ideas and things, trajectories, to see whether you could figure out whether there were polyploids going along certain trajectories in that space and ending up in a particular point. Those are the kinds of things I think we may be able to get closer to doing with the kind of the phased haplotype genomes that we have now. Now, the problem is that you need evidence, right? What's the evidence you can do? You can make lots of crosses. You can figure out with the techniques to look at progeny now and segregation ratios. But as I said, that gives you only a snapshot of what's happening now. True, that's also true for genomes. You sequence genomes, but pangenomes and all the rest of that give us a lot more richness to be able to think about what the distribution actually is out there and how that corresponds to the categories that we've already used. Taxonomy being one of them. There's nothing wrong with having taxonomy as one of the criteria that you're using for that. And if you could relate it to morphology and physiology and the rest of those things, how wonderful would that be too? But we're not there. Adrian.
Really fascinating talk. You said that most cell types increase in size when they're polyploid, but you implied that some don't. What doesn't increase in size as polyploid?
Well, there's the Katagiri paper from Tsukaya's lab, right? That's the one example that I know of off the top of my head where they said that, what was it, epidermal pavement cells do increase in size, but they, I forget which cells. The mesophyll doesn't, right? And, well, so increase in size, right? I mean, we don't need to get into that, but there's a lot going on with what controls cell size. But for those of you who haven't heard older seminars from me, Adrian, I once went to Adrian a number of years ago and said, Adrian, I'm interested in polyploidy's effect on biology, cell biology, and I know that it increases cell size, but there's some cell type thing here. What's a cell type? And I expected the answer from a cell biologist to be, here's the definition of a cell type. The answer was, cell biologists don't know and don't agree what a cell type is. My response was, what have they been doing all those years? Since 1846 or 1646, and with all the money they get from biomedical science. Okay, anyway, but it's a tough question. Tom.
Is there an energetic cost to polyploidy?
Yeah, it's a good question.
What are the advantages? What's the payoff?
Yeah, so there's some wonderful work on snails, of all things, by Maureen Nyman at University of Iowa, looking at diploid and polyploid New Zealand snails. And New Zealand snails at that. And what she is seeing that there is a cost, an energetic cost, because of the phosphorus and nitrogen involved in even just making a genome, right? But then also transcribing it as well. So then there are studies also, kind of long-term ecological studies in England where they've grown plants over many, many 100 years or a couple hundred years, something like that, on either fertilized or unfertilized soils. And what they look at, when they look at the kind of plants, the species that are there and the genome sizes of them, the genome sizes on average are larger of the plants growing on the fertilized than the unfertilized plots. And so people are seriously thinking about what the costs of polyploidy might be. There are other costs too. I mean, in terms of just getting them started, being able to actually compete against diploids in a natural population as a minority cytotype, for example, because they're going to be the minority to start with. This gets us into all kinds of population genetic questions too, which are really complicated for multicellular eukaryotes where effective population size is small and genetic drift is much more of a force than selection. So the selection coefficients have to be quite high to actually go either way. So there's a lot of stuff that goes into thinking about how polyploids compete or don't compete. That help?
It's
some of the answer.
It sort of reflects
Yeah.
what what I've been thinking.
Okay. Yeah. Okay. Someone over there. Yeah.
Hi, very good talk. I learned a lot about the polyploids. So my question is the polyploids could bring lots of benefits for plants, but it also takes more energy and time for plants to duplicate some information during mitosis and meiosis, and
Right.
also the gene loss will happen more easily during this time. So do you think plants need to make a balance between polyploids, how much number of polyploids, and avoid gene loss or save energies?
Right. So essentially, you're asking somewhat the same question again about what the cost of polyploidy is, and whether that's a cost of the raw materials for duplicating the genome, the rate at which the genome is being duplicated too, because one of the nucleotypic effects, other than just cell size, is also the rate or the time to completion of mitosis and meiosis is longer when you have more DNA to replicate. So that can be a cost as well. And certainly in some environments, that's a problem. There's a correlation, for example, between genome size and whether plants are living in high Arctic environments where they have a very short growing season, or high altitudes. So this is another way in which maybe polyploids are not going to be found there, where there's a constraint on genome size. Genome size in general and polyploidy are related to one another. Obviously, and whenever you duplicate a genome, you increase the genome size, but they're independent as well. There's a lot of overlap between diploids with large genomes and polyploids with smaller genomes. So it's hard to generalize completely about some of those costs and things as well.
Could you check?
Very short comment.
Any question?
Oh, thank you. Yeah,
To
go on.
what you just said, Jeff, you're certainly aware of that. There were some studies from Sally Otter's lab about 15 years ago showing that most polyploids actually go nowhere. There are very few that I know it's been controversial.
Well, can you come up and see if you can find us while I talk to that question? So success has been something people have debated in the polyploidy literature ad nauseum. I hate it. I just hate it. And the reason is because nobody ever defines success.
That's the reason
rarely
I didn't
do.
mention
Yeah,
it in
yeah.
the first place.
So is success. So here's my question. I've got to go, don't I? I've got to get out of here. Yeah, the boss is telling me to leave. I want to leave you with a thought on that. Do you consider Homo sapiens to be successful? Okay, and the reason I ask that question is this, because some of the measures of success that people use, the main one that people use is, has it given rise to new species to increase the lineage and there are more species there? Well, humans haven't done that, not as far as I know, anyway. Well, maybe the White House, I don't know. That's, well, but yeah, so there's that. But there's also another sense of success is just, have you dominated your ecosystem? Well, humans have done that, but we've only been around a short period of time. And a lot of polyploids are very successful locally. Now, if they don't give rise to new species eventually, who am I to judge that? I mean, a human has been around a short period of time. But anyway, so how you define that is a real problem. You can be very successful in the short term. And is that success? Polyploids do it.
Jeff, there's one question here. There's comments, but
Okay.
There's only a question.
So if a pairing control switch exists in polyploids, is it more likely to be triggered by sequence divergence, structural heterozygosity, or epigenetic remodeling? Well, I think the PH gene, Wojtek left, that's too bad. The PH gene, pairing homologous gene in wheat, is a gene that operates epigenetically, I think. It's a complex situation, and that's what gives preferential pairing in wheat. Why the wheat genomes, even though they're very recently put together, they are disomic, perfectly disomic because of this one gene. You knock out the gene and you get homologous pairing. So it can be genic, but the control, the line between genic and epigenetic is, I think, fungible, if nothing else. Right? Thank you all.
This has been a production of Cornell University, on the web at cornell.edu.